In 2026, researchers X.Y. Song, Y. Shah, and their team published a paper in bioRxiv titled Antiviral Reverse Transcriptase Primase Synthesizes Protein-Templated DNA. For decades, the rules of biology have been clear. If an enzyme called a reverse transcriptase, or RT, is going to manufacture DNA, it needs an existing strand of RNA to use as a template. It needs a blueprint to read from. But out in the microscopic wild, bacteria are locked in a constant lethal arms race with viruses known as phages. To survive, bacteria have weaponized these RT enzymes, turning them into potent immune defenses. Which brings us to the anomaly at the center of Song and Shaw's study. They looked at a specific class of multidomain defense enzymes called DRTs. One of these, DRT7, has both an RT domain and a primase-polymerase, or PP, domain. But it completely lacks the non-coding RNA template that reverse transcriptases usually require. If a bacterial protein doesn't have a genetic blueprint to read from, how is it manufacturing the complex, lethal DNA needed to fight off a viral invasion from scratch? To find out, the researchers first needed to measure DRT7's defensive capabilities. They introduced the enzyme into E. coli and ran plaque assays and growth curves against a highly diverse collection of phages known as the basal panel. The presence of DRT7 caused a massive million-fold reduction in phage survival. The infected cells underwent growth arrest, sacrificing themselves through a process called abortive infection to stop the virus from spreading. By analyzing phages that managed to mutate and escape the defense, the team discovered that DRT7 only activates when it detects a specific viral transcriptional regulator entering the cell. To identify the specific biochemical output DRT7 produces when it activates, the researchers moved to in vitro DNA synthesis assays. They isolated the enzyme, supplied it with DNA building blocks, and tracked the resulting reactions. The synthesized DNA was exceptionally long, averaging 1,800 base pairs. On an SDS phage gel, it caused an upward mobility shift, and it proved highly resistant to exonuclease 1, an enzyme that normally degrades the free ends of DNA strands. This diagram represents the sequencing data gathered using Illumina and PacBio SMR technology. You can see the sequence architecture. The DNA is constructed of strictly alternating palindromic blocks of repeating adenines and thymines, poly-A and poly-T tracts. Combining the nuclease resistance with these sequencing patterns led the researchers to a distinct conclusion. The DNA is covalently linked directly to the protein, and the long, alternating tracts force the sequence to fold back on itself into stable hairpin structures, eliminating any free ends. To understand how a single protein physically builds this specific structure, the team used cryo-electron microscopy. By freezing DRT7 in action, they captured its 3D architecture in both inactive and active states, revealing the mechanics of the synthesis. This schematic shows DRT7 in its initial closed dimer state. Instead of reading an RNA template, the RT domain uses a highly specific arginine-rich pocket to grab a single thymidine and attach it directly to an amino acid residue called Y682. From that single starting point, the RT domain continues to add more thymidines, building out a short continuous poly-T sequence. The critical trigger occurs when that chain reaches exactly 11 nucleotides. The physical strain of the growing DNA forces the protein's middle domain to snap into a dramatic 180-degree rotation, transforming the entire structure into an open, active dimer. That physical transformation initiates the iterative handoff. The newly synthesized poly-T strand shifts out of the RT active site and moves over to the newly repositioned key P active site. Here, the PP domain takes over. It utilizes that RT-generated poly-T strand as both a structural guide and a primer, assembling a complementary sequence of adenines via standard Watson-Crick base pairing. Once the poly-A tract is complete, the end of the strand is handed back to the RT domain for another block of thymines. The DNA loops back and forth between the two domains, rapidly spinning out into that massive 1,800 base pair hairpin structure. This cycle proves the existence of a biological engine previously unknown to science: protein self-templating, sequence-specific DNA polymerization. Back inside the host bacterium, the rapid production of this massive, custom-built DNA tangles up the cellular machinery. This structural chaos successfully triggers the cell suicide we saw earlier, stopping the phage fission. We have long assumed that creating complex, sequence-specific DNA strictly requires a pre-existing nucleic acid template to read from. DRT7 proves that this is not always the case. By understanding this iterative handoff, we can potentially harness DRT-like enzymes for the programmable de novo synthesis of complex DNA structural elements and nanomaterials, entirely without a traditional blueprint. We owe this leap in our understanding of prokaryotic immunity to the careful work of Song, Jia, and their colleagues in their 2026 publication. As we look at the summary slide of their findings, remember the key takeaways. DRT7 provides broad-spectrum immunity by triggering an abortive infection mechanism, and it only activates in the presence of viral transcriptional regulators. Upon activation, the enzyme synthesizes an 1800 base pair palindromic poly-A and poly-T DNA structure. The RT domain initiates this process without an RNA template, relying entirely on its specialized arginine pocket. The elongation of that initial strand to 11 nucleotides forces a massive 3D structural shift, driving the iterative handoff mechanism that bounces the growing DNA back and forth between the RT and PP domains. The microscopic arms race has revealed a biological reality we hadn't anticipated. Down at the molecular scale, bacterial machines are building their own genetic blueprints from scratch to ensure their survival.